meet you. So thank you for inviting me. So I just share my screen, I guess. So here, and yeah. So I'm also at the University of Poitiers as an associated researcher, and I'm basically a paleontologist and an anthropologist, and I'm working on the ear and in its evolution in primates and more largely in mammals. So yeah. So and now I'll try to share my presentation more. Yeah, here. So now you should be able to see just the presentation. So here today I'm going to talk about the evolution of the mammalian ear, and especially an evolvability hypothesis. So this is a hypothesis that I published with other researchers at the University of Vienna, so Catherine Pfaff, who is here, and Nicole Gonsra and Philippe Maître. And the aim of this hypothesis is to explain how comes that the mammalian ear evolved. So first, just a reminder about the mammalian ear. You know, it's involved in multiple functions with auditory system for hearing and the vestibular system involved in balance, posture control, gaze stabilization. And it has several elements in 3 parts. So on the you have the external ear with the auricle, which is external, and the external auditory canal. Then it's separated to the middle ear by the eardrum, and you have the middle ear, you have a cavity with 3 auditory ossicles, and then the inner ear where the sounds are detected on the cochlea, and the head movements are detected by the semicircular canals. And this organ is integrated, it's very tightly integrated in the temporal bone, so spatially, functionally also for the audition, because you need the outer, middle, and inner part for audition, and also developmentally. So, for example, the ear has a multiple number of developmental origins with at least 4 different tissues. So on the A panel corresponds to the lateral view of an embryo with the head on the upper side and the dorsal part on the left. And so, in fact, the ear comes from the 2 pharyngeal arches, PA1 and PA2. And these pharyngeal arches have give different elements. So the first pharyngeal arch gives rise to 2 middle ear ossicles, the malleus and the incus, M and I on the right side figure, whereas the second pharyngeal arch give rise to the stapes, so S on the left and right figure. Also, you have the inner ear, so in green on the right side. So this comes from a part that is ectodermal tissue that is located between the third and the second pharyngeal arches. So you have multiple developmental origins, and all these things are integrated to give one functional organ, the ear. And also, when you look at mammals, you have a huge disparity of size, from the tiniest bats and rodents to the whales and the elephants, a disparity of body plans, a disparity of shape, a disparity of environments with aquatic, aerial, terrestrial, subterranean, arboreal species. You have a diversity of ecology in terms of locomotion, very slow, very fast, jumping, swimming, and also in terms of hearing, some that are able to hear ultrasons or infrasons or to do echolocation. So there is a huge mammalian disparity. And so the question that arises is that the mammals, which are predominantly a nocturnal radiation, especially at the base of the evolution, they rely on hearing. And so how comes that this group that relies on hearing can manage to occupy such a huge diversity of ecological niches, and that's the tightly connected part of the ear, which is just many different parts completely connected, how can they adapt independently to these diverse functional and environmental regimes? And to answer this question, one way is just to compare mammals with other groups. So for example, with birds and lizards. And when you look at the ear in birds or lizards, you see some difference. First, for the inner ear, so on the left you have the birds and lizards, right, the mammals. And so you can see that the inner ear, the cochlea is very short and it's not coiled in birds and lizards, whereas in mammals it's coiled. Also for the middle ear, you have a cavity like in mammals, but there is just one bone in birds, whereas there are 3 bones in mammals. Also, the eardrum is connected to the skull by just 1 bone in mammals, the tympanic ring or the tympanic tube, whereas in birds and lizards, it's connected to different bones, but it's not the same bone. And also, generally, you don't have an external ear in birds and lizards. So there is a lot of differences. And when we try to see if there are homologies between mammals and non-mammals, for example, you see that in fact in mammals, so on the right side you see a mouse, so you have in red the malleus and the incus, and in fact these bones are homologous respectively to the quadrate and the articular bones in birds, which are bones of the jaw joints. Also the green bone in mammals is the tympanic ring, so where the eardrum is attached, whereas in birds it corresponds to the angular bone, which is a bone of the jaw. When you look at the stapes of the mammals, it corresponds to the comella of the chicken, so it's an ear bone. But you see here that most bones that are involved in the ear in mammals corresponds to bones of the jaw joints in birds. And so this is a huge difference between mammals and birds. And how comes that this mammal developed such a huge difference compared to other tetrapods or vertebrates? And in fact, when you look at paleontological evidence, you see that the very basal species, so the cynodonts that are a group in which the mammal branch, this is some kind of reptile, but not mammals. You have the stapes, which is a bone of the middle ear, and you have the 3 bones, quadrate, articular, and angular, that are parts of the jaw joints. And so in this situation, you have the hearing module and the chewing module that are combined, that are joined. So there is not a strict separation in the chewing and hearing function. And when you look at more recent species, for example, Morganocodon, Leiacodon, then you see that you have some changes in the shape of these jaw bones that becomes smaller and smaller. And then at some point, there is a decoupling. And so that at the end, you see that in species like the opossum, which is a mammal, you have a complete decoupling between the middle ear ossicle and the jaw. So there is a complete decoupling between the hearing module and the chewing module. And this decoupling, so this transformation from the jaw joints bones to middle ear ossicles, it happens several times independently during the mammalian evolution. And so this is a major transition in the mammal evolution. And so how to explain this transition? So there are several hypotheses that were proposed historically. So the first hypothesis is that when you have more bones in the ear, you have a more complex ear with more elements, so you have new hearing abilities that can be developed, such as ultrasonic hearing, infrasonic hearing, or echolocation. So a second hypothesis is more that you have a selection on the jaw and how the chewing module. So when you have a less complex jaw joint in mammals, you can do some other kinds of chewing movements. And especially you can do sharing masticatory movements, and you also have stronger jaw. So this is the second hypothesis. And the third hypothesis is that because of the decoupling of hearing from mastication that you have in mammals, then you can have the selection on two different functions separately. And so you can evolve independently these two functions. So these are the three hypotheses. So either a selection just on hearing, or just on chewing, or a selection that can happen to both modules because of the decoupling. And with my colleagues from the University of Vienna, we proposed a new hypothesis. This is the evolvability hypothesis. And this hypothesis is that with the incorporation of the bones of the primary jaw joints in the mammalian ear, you increased the ear complexity for mammals in terms of development, in terms of genetic control, in terms of regulation. So for example, you have the malleus and the incus that are controlled by, that have a developmental origin in the first pharyngeal arch that you don't have in birds or in lizards. So you have a more complex development, so more possibility to for the evolution to play with this evolution and with the variability of the ear elements. And because of this increase in ear complexity, then you have more degree of freedom for the independent adaptation of the different functional units of the ear. And this increased degree of freedom for the adaptation would have led to the adaptive diversification of mammals. So very early in their evolution, and especially after the dawn of dinosaurs, when they died at the end of the Cretaceous, there were a lot of evolutionary niches that became empty. And so the mammals could very quickly go into these niches and adapt to these niches thanks to this ability to evolve new ear shapes. And this would have given a long-term fitness advantages for mammals. And what are the prediction for this hypothesis? So first, that for mammals, compared to other clades or vertebrates, you would have an increased modularity of the different elements, and hence a reduced integration, and also a higher multivariate dimensionality of ear shape. The second prediction is that you would have a greater evolutionary rates and disparity in mammals compared to clades of similar age. A third prediction is that because of this higher evolvability, you would have a greater potential to develop, to innovate, and to have evolutionary novelties, and also for convergent evolution. So similar evolution, similar shape in response to the same environment. Also, you would have a higher correlation in mammals compared to other groups between ear morphology and variables like ecology, behavior, or audition. And finally, this increased evolvability of the ear in mammal, because of this higher complexity, would lead to a higher susceptibility to developmental instabilities and disorder, because if you have more complexity, it's easier to have problems during the development. So this is the prediction of our hypothesis. But does this, is it seen in real life? So when we look at the outer ear diversity, so first we have an innovation in mammals. This is this external ear with this pinna. So here, for example, you have the diversity in primates. So this is one innovation. This is present just in mammals. Also, when you look at the bone that is supporting the eardrum, you have a single bone that is derived from the angular bone. And this is also an innovation in mammals. And you have also a lot of diversity because, for example, here in primates, sometimes this bone takes the shape of a ring, sometimes it's a tube. So it depends. So you have an innovation and diversity. Regarding the middle ear, you have some innovations. So first, the two new middle ear ossicles, but also in terms of shape, you have a huge diversity of shape. So for example, you have a huge head of the malleus, so for the C in some species, which is an adaptation for to detect some vibration for subterranean species. Also, you have different types of middle here, so the way the shape of each ossicle and how they are arranged and how they function one relative to the other depends. And you have some convergence. So, for example, the A type is a microtype, and it's very present in very small mammals. And it's adapted to detect high frequency. You see it, for example, in bats. Whereas the D type, which is a freely mobile type, is present very frequently in all kinds of subterranean mammals, and it's adaptive to hear low frequencies due to ground vibrations. Also, you have the middle ear cavity that can have very different shapes. So in red, this is the auditory bulla. So this is a huge, it can become very huge in some species. So this is also specific for mammals. Regarding inner ear, you also have a lot of diversity. So for example, on the left, you have the diversity in cetaceans. You have a very developed cochlea, and compared to the small semicircular canals. And this developed cochlea is an adaptation to infrasonic or ultrasonic hearing. Also, you have a huge diversity in other kinds of mammals. So for example, A corresponds to ornithorhynchus, a B to opossum, and all other shapes of cochlea correspond to other placental mammals. And you see that you have a huge diversity in the length of the cochlea, in its width, and the number of coils. Also, you have different novelties. So here, each number corresponds to a novelty in the different groups of mammals during the time. So from a very short and not coiled cochlea to longer cochlea, for example, in monotremes, but still not coiled in ornithorhynchus, whereas you have a hugely coiled cochlea in placental mammals. Also, in terms of the structure of the corti organ, you have many different innovations. So this is for vertebrates in general. And for the mammals on the left, you have innovations in terms for the outer and inner hair cells. And you have a thinner basilar membrane compared to reptiles or birds. So you have a lot of innovations and a lot of diversity. And you don't have just a lot of innovation and diversity. Also, this diversity allows mammals to be more adapted. So here, for example, you have an example in some rodents, in some subterranean mammals, like moles or shrews. And here it's a plot of middle ear ossicle shape. So malleus in purple, incus in yellow, stapes in red. And the color corresponds to different kinds of locomotion. And you see that, for example, the subterranean species have some special shape, whereas the more terrestrial species have another shape. So in mammals, you have a very good fit between the shape of the middle ear ossicles and the ecology. Whereas in non-mammal case, so for example, in turtles, so here the different colors and symbols correspond to different kinds of turtle ecologies, and you see that there is not a very consistent pattern separating the different kinds of adaptation in turtles. For the inner ear, you have a similar thing. So for example, in squirrel-related rodents, you have different shapes between flying species, flying squirrels, subterranean species, and arboreal species, and terrestrial species. So you have some separation. Whereas in birds, you have the morphology of the ear. It's much less related to the type of ecology. So, for example, the yellow points are flightless birds, the red points are hunting birds, and so you don't see a clear pattern. In fact, in birds, the ear shape is more related to brain size and brain shape compared to the ecology. So for both middle ear and inner ear, the ear morphology is more adapted in mammals compared to other groups. So to conclude, so the transformation of the middle for the mammalian ear, this is a very important transition during mammalian evolution. And you have all hypotheses, but here we propose a new hypothesis, the evolvability hypothesis, that states that because the mammals have much more constituents, you have much more possibility for the variability of the different elements, because you have much more developmental, genetic, and regulatory pathways to create these elements. And so it allows a more easier adaptation of each element to different environments. So the elements that support this hypothesis are the higher morphofunctional fit in mammals compared to other groups, the paramount number of evolutionary novelties in mammals, and the convergent evolution that is very frequent in mammals, for example, for subterranean mammals, for aquatic mammals, for ultrasonic hearing, etc. And so there remain things to be tested. First, so here the only thing that I show is mainly qualitative studies, or very studies dedicated just on mammals or just in one group. And one thing that would be good to test this hypothesis would be to compare really with the same data set mammals to a clade that is of similar age. So for example, placental mammals versus birds, and to compare variability, integration, modularity, morphofunctional correlation between these two groups. And we expect that in mammals you have more integration, less integration, sorry, but more variability, more modularity compared to the other group. And so this is what we are doing with my colleagues as part of our projects in Vienna. And also there is another perspective of this hypothesis, is that this more increased complexity of the mammal would that would lead to developmental instabilities that would explain, in fact, why the ear is so unstable in mammals, and especially in the human species. And maybe if we would understand better how it's integrated and how this evolvability has an impact on developmental instability, then it would help us to tackle some ear disorders that are related to more for morphological defects of the ear. And so on these words, I would thank you all for your attention, thank the organizer for inviting me, and also thanks the following colleagues of Philippe Minterker, Nicole Grunstra, and Catherine Pfaff for their involvement in this project and the funder. So thanks.
Well, thank you very much. Very nice talk, even though I'm not a specialist. So we have about 8 minutes for the discussion. If you allow me, I will start by a very general question and then give the question to the other people. In the geological times that you have shown, of course, the changes have been very slow and the adaptation could be very slow, but now animals and mammals are sustaining very rapid changes due to increasing stress, due to the anthropogenic activities, increasing temperature, maybe due to global climate change, due to different sources of noise, not sound, noise actually due to the anthropogenic activities as well. So do they have time to adapt? Is it possible that they adapt really that fast? And is there any threshold beyond which they will just die?
So in terms of adaptation, I think, yes, there is a lot of possibility to adapt. And in fact, there is some intraspecific variability, and we are conducting a species on the inner ear of humans compared to rodents, and we see that, yeah, there is a lot of variability, and this variability between individuals, but also during the development, allows some kind of plasticity to adapt to different environments. But it's difficult to say if there is really a threshold beyond which it's no more possible to adapt. But I would say that because mammals have such a complex ear, they are more susceptible to adapt compared to snakes or to birds or to lizards in terms of hearing abilities to anthropogenic related changes.
Okay, so some other questions?
Yes, I have a question. Fascinating talk. I love a lot. Really, evolution is so fantastic. Now, I have just a question on elephants.
Yes.
So elephants, they can hear, as far as I know, they can hear a low frequency. And I'm curious, what is the adaptation? I see the big external ear. What is about the middle ear, the inner ear? I heard about they have also sensation in the leg, so to hear sound from kilometers. So this is a fascinating animal. You have some data to give me.
So I'm not sure for elephants because it's not so much studied species, in fact, just because it's difficult to have scans of such huge scale. But I think that there are some adaptations in terms of cochlea shape, that the basilar membrane is I think thicker and a bit larger to hear lower frequency. And also regarding the middle ear ossicle, I think the malleus has a special shape that changes a bit the balance of the ear ossicles one relative to the other, and so it allows to detect slightly different sounds. And I would say also maybe there are some adaptation to hearing, not through the middle ear ossicle, but directly through the bones. But I'm not very sure about this.
Thank you very much. Fascinating. Thank you.
Other questions?
Yeah, if I may, because follow up exactly on the same question, I have a comment and same time a question for Anne. Just to answer the question of Jean-Luc, you know, even in mammals, there are evolution. There are bats and there are humans. Bats are one of the highly evoluted species. And we in mammals, the hair cells in the cochlea, they are small, they have shorter stereocilia, which are more rigid. And as you go to the low frequency, they had more longer hair cells, more flexible stereocilia. And if you look at in bats, which requires high frequency hearing, they all have very short outer hair cells and very rigid stereocilia. So I would guess that in elephants whose hearing requires also low frequency, the hair cells will be longer, more flexible, stereocilia will be thinner and longer. I have a question to Anne. I like evolution, so which means also I like the talk and the comparison. What's important about comparisons is that you learn from differences as much as you learn from similarities. And you mentioned the three hypotheses, for instance, how we load the decoupling between mastication and hearing, and you mentioned the three hypotheses. And I know that in life, we always like either to see it as black or white, but we forgot all the shade of gray between black and white. So I think the three hypotheses, in fact, can apply. It's a mix of everything. I mean, we need some of the ossicles in the jaw because of the needs there, and they are sufficient for low frequency hearing. But as you move on from water to air, so you need you don't need to rely on the ossicles in the jaw. So evolution doesn't like waste, so she took at some point we don't need that for the jaw, so they are taken out or harnessed by the inner middle ear. So how that fits with the evolvability hypothesis, the fact that we have a mix of everything.
In fact, all four hypotheses are not contradictory, because I guess, yeah, you can have all three of them. So you have a decoupling, and then you have selection for hearing abilities and selection for masticatory abilities. In some groups, this is the selection for masticatory abilities that is stronger, in other for auditory abilities. And evolvability is just here to increase the ability to respond to these different selections. And I guess it's just the evolvability hypothesis, just some hypothesis that is underlying all of them, because without evolvability, in fact, you cannot respond to selection anyway. So yeah.
Thanks for your talk.
Okay, a last question, very short. Otherwise, I ask it because we are in the session on interdisciplinary, transdisciplinarity. If you would need one or two more expertise, which one would it be, and to do what? To go towards interdisciplinarity.
So yeah, I would say here that it's so interdisciplinary, because here to test this hypothesis, you need elements from many things, from paleontology, from embryology, from just comparative anatomy. So it's, yeah, the hypothesis itself is based on some interdisciplinary work. So in my current work, in fact, I'm working just with extant species. So and just in adults. So if I'd like to add some interdisciplinary, I would add some developmental studies in some specific model species, and also some studies of some fossil species in a very specific group, just to check that so if we find a higher viability in mammals compared to birds, for example, just to check that this is also true for fossil mammals compared to fossil birds. So,
okay, thank you very much.
I have a question.
Yeah.
Sorry, probably naive question, but I was wondering if your hypothesis could fit with the brain evolvability.
Yes, in fact, I have a side project where I try to understand if during human evolution, where the brain becomes larger and larger, and it takes more and more space, then it reduces the size available for the ear to evolve. And so you have, maybe this has an impact on how evolvable the ear is, and maybe it's in the human lineages, or more generally in primates, where the brain is very large, you would have a smaller evolvability of the size in the ear, but maybe that would be compensated by a larger evolvability of the shape compared to other mammals with smaller brains.
Okay.
Okay,
thank you.
I can make a comment. Maybe we compensate this slower size of the ear by larger neural networks interpreting the
also, of course too, yes. So I was just speaking about the morphological aspects, but of course for hearing all the neurological and all the soft tissue and interpretation is very important, of course.
Fantastic talk.
Thank you.